Anthropogenic light pollution comes in two main forms. Point sources like street lights, house lights, or lit windows have intense impacts on their immediate environment. When that light is directed or reflected up into the sky, it becomes a diffuse haze visible for miles and miles, which we call sky glow. Models built from satellite imagery, like that shown on the right, show how sky glow diffuses out from centers of human development. In this map, black and gray represent pristine night skies, and as you can see, over half of the US and 23% of the Earth is unnaturally bright at night. And our nights are only getting brighter over time, nearly twice as bright in just the past decade. Over half of all animals, most animals, are active at night, and our light is completely transforming their world. They struggle to cope with this change in particular because it has no natural analog. I've spent a lot of time studying whether fireflies will survive, given how difficult we're making it for them to communicate. And I'm going to talk about that later on. But more recently, I've turned my attention towards moths. Moths are underappreciated architects of our ecosystems. As caterpillars, they shape our farms and forests through herbivory. As adults, they shape our farms and gardens through pollination. And along the way, they feed the birds, frogs, and other creatures that people actually like. Like all insects, moths are affected by sky glow and point sources of light in many ways. I don't have time to talk about all of them here today. And I know that many people here are interested in daily and seasonal timings or chronoecology. There's a lot to talk about there, which maybe we can do afterwards. But for now, I just want to talk about this weird thing that moths do, where they spend entire evenings, a significant chunk of their short lives, banging themselves against lights over and over again, circling them or sitting stunned beneath them, instead of working to perpetuate their species. We still don't really know why so many moths do this, but it's very clearly an accident, and it's a costly one. I want to understand how moths are doing in response to artificial lights, but this turns out to be a very difficult problem to solve because we use artificial lights to understand how moths are doing. Entomologists use light sheets and light traps of one form or another to systematically survey the abundance and diversity of insect communities, and have for a very long time. These lights are usually black lights, which have a strong UV component, either mercury vapor, fluorescent, or here's a photo of me doing a very non-systematic survey last summer using the Lucky LED. When we don't see moths at lights, we get worried. In recent years, scenes like this have sparked fears of an insect apocalypse, which are heightened by anecdotes about not seeing insects caught in car headlights, plastered on lit windows, or swarming around streetlights. When we first started using artificial light to survey moths around the turn of the 20th century, Boston Common at twilight looked like this. You can see a few gas lamps. They're not all that bright, and they wouldn't have been on all night. And in fact, in 1900, the entomologist Sherman Denton wrote about this. He wrote about visiting the new electric lights in Washington, D.C., and said that he had made a splendid collection of the moths of that region. And not only that, met several entomologists who were attracted to the lights by the abundance of specimens. Today, Boston Common at twilight looks like this. And at these lights, you're not likely to find very many moths or entomologists. So we used to see many moths at streetlights, and now we don't. We typically assume that a light trap catches a constant proportion of the surrounding population. In this simplified example, that proportion will be 50%, and moths not caught by the lights will be shown in gray up there. These kinds of data show that we've gone from 6 moths to 2, a defined as 66%. This is the apocalypse scenario. It's very scary. One alternative explanation, however, is that light-attracted moths today are spread among more lights, and that each individual light is less attractive than it used to be because it doesn't show up as brightly against all of our light pollution. In this scenario, the moths are still there at lights. It's just that the light traps used to monitor them are being outcompeted. Another alternative explanation is that moths today are simply less attracted to light than they used to be, because flight to light behavior is highly maladaptive, and they've already had several hundred generations in which to figure this out. In this scenario, the moths are still there, just not at the lights. To evaluate which of these three scenarios is responsible for the patterns that we see, all we have to do is compare what the light traps are telling us to what other traps are telling us. And I thought this would be simple enough going in, but soon discovered that people rarely use other methods of systematically catching and counting moths, except in the world of agricultural entomology, where the corn earworm in particular, a highly destructive pest species that targets corn, tomato, cotton, and hemp, has been tracked extensively by the USDA and affiliated cooperative extension programs for decades. There are two modern long-term monitoring networks that I'm going to talk about today. One is run by folks at the University of Delaware and consists of 11 farms, and one is run by folks at Rutgers University and consists of 7 farms. Crucially, each of these farms has a pair of corn earworm traps, one blacklight trap and one pheromone trap. This redundancy is meant to increase the chances of detecting outbreaks before they become serious, so that growers know to spray. But both of these networks also maintain high quality long-term records. So I reached out to the people involved with this question I had that could be answered with their data, and thus began a beautiful corn pest collaboration. My student Ian Battles, an undergraduate at Northeastern University, conducted the analyses I'm about to present. So here is one of the Delaware farms in the middle there. This is near Bridgeville. The blacklight trap is located at that point, and the pheromone trap is there, less than 1 kilometer away. Each pair of traps at each farm are monitored throughout the summer, and average corn earworm count data are recorded every few days. At farms in Delaware, this has been going on since 1998. The same blacklight with the bulb replaced each year, the same brand of pheromone trap, and the same general location. Here are the raw data for Bridgeville, where the height of each line represents the average moths caught per night across the summer months. For each trap in each year at each farm, we lump these together to generate a single value, the index of abundance, where a bigger index means that more corn earworms were caught in that trap at that farm that year, and each year has counts for multiple farms. The pheromone trap data look like this. You can see there are no major changes over time, except for a bit of a dip around 2010, corresponding with the popularization of GMO corn. Corn earworms went down a little bit, very rapidly evolved resistance, and today seem to be doing pretty well. So I'm going to show the blacklight trap data in a second, but first I just want to say, if blacklight traps have not been compromised by light pollution, if they still work as well today as they used to, we would expect this line and the line I'm about to show to be parallel. The overall average could be higher or lower because these traps catch different numbers and kinds of moths. Pheromone traps are highly targeted, but only catch males. Blacklight traps can catch non-target species of both sexes, and so on. So the intercepts could differ, but the slopes should not. The percentage of the population caught by each trap should be constant over time. This is a foundational belief. However, the blacklight trap data look like this. They start at around the same place, actually, but diverge extremely rapidly. These are just the data from Delaware, where the monitoring network has been running since 1998. The New Jersey monitoring network has been running since 2013, and it turns out that 10 years is enough time for these lines to diverge significantly in the expected direction. Here, the pheromone trap counts are on the rebound, while the blacklight trap counts stagnate. And if you look at only the most recent 10 years of the Delaware monitoring network right next door, the situation looks almost exactly the same, even though these are completely different monitoring networks run by different people in different places. It's not necessarily surprising to me that 10 years is enough time to see a significant decline in blacklight trap effectiveness, because as I said earlier, the night sky has become twice as bright in just the past decade, such that on average, if you could see 250 stars in the sky at night as a child, today you can see less than 100. And that is thanks in a large part to the growing popularity of super cheap, super bright LED lighting. So I suspect that many people in this room are yearning to see a generalized additive model. There it is. It shows that for the first 12 or so years of the monitoring network, blacklight traps and pheromone traps caught about the same numbers of moths. They only really started to diverge again around 2010, which is also around the time that Delaware began replacing older streetlights with modern LEDs. which might have increased light competition. If the surrounding light environment is compromising black light traps directly, we might expect greater divergence between trap types at more light polluted farms. So we use satellite data to estimate environmental light levels in a 1 kilometer buffer around each trap location and looked at how that affected the difference between trap types. So on this plot, a greater difference indicates greater divergence between the two trap types. And the x-axis is one of the many, many ways of measuring light pollution. We did not see a strong relationship, which could be because this phenomenon has nothing to do with light competition and is all about rapid evolution. Because this species is highly migratory and well mixed, and in fact repopulates Delaware and New Jersey each year from overwintering sites in the South, it's entirely possible that the entire species is evolving and learning as one. And we'll come back to that. But it's also worth mentioning here that satellite data have their own biases and in fact are also getting worse over time. As I keep saying, much of the globe is currently undergoing a transition from older lighting types like high pressure sodium, shown there on the right, to blue rich LEDs. On the left, these are photographs of Milan from space taken before and after a streetlight conversion. It looks like it gets a bit darker, but on the ground it's much brighter. And that discrepancy is partly because no satellite in orbit at the moment is capable of measuring light below 480 nanometers to the left of that line, which means a bunch of this added light is getting missed. Not only that, thanks to astronomers, new lighting installations are more likely to be shielded to prevent that light from going up into space, where it can interfere with astronomical observations or get picked up by a satellite. But I don't know how much it matters to a moth whether a light is shielded or not. Shielded lights can be hugely attractive to insects on the ground, and gas station lights are a good example. So we shouldn't necessarily expect to be able to see what a moth sees from space. That's all I'm going to say about satellites, lest we forget who we're talking about. Looking at all of the data from the Delaware Monitoring Network again, we can see that the corn earworm seems to be doing pretty well overall. I would not say that these guys are of conservation concern. However, if we only had data from blacklight traps, we might have a very different impression. Let me highlight now that these data are on a log scale. The difference is extreme. Blacklight traps catch so few corn earworms today that a lot of agricultural entomologists don't bother using them. But insect conservationists use them because they're all that we have. For most moths of conservation concern, we only have data from blacklight traps, but these data are conflated, which makes things complicated. To summarize, the blacklight traps in Delaware used to catch many moths and now they don't. Is it because the number of corn earworms has changed? No, because the pheromone trap counts remain high and the growers still have to spray their corn. It could be because the moths choose to fly towards the LED streetlights instead of the blacklight traps, or because these and other nearby light sources make it harder for them to see the blacklight traps. Or it could be because the moths that fly to streetlights and other light sources frequently die before they can pass on their light-attracted genes to the next generation. Perhaps strong selection against flight-to-light behavior has led to rapid evolution. If that is the case, then when we rear these moths in a common garden and test them in the same location, we might expect to see a pattern like this, with rural moths today less likely to fly to lights than their historical conspecifics, and urban moths even less so, as they have had more generations in which to adapt. Altermat and Ebert found this pattern in rural and urban populations of small ermine moths, but no one has compared historical and modern behavior in this way. I suspect that one reason is because while historical reports of moths at lights are common, concrete percentages are very hard to come by. But in 1969, the distant past, Paul Stewart and colleagues measured the percentage of corn earworms that flew towards a black light at different distances. So we have replicated this experiment as perfectly as possible 55 years later, using the same moth species, the same 15-watt black light, and the same experimental design, which involves these massive 8-foot-long tunnels that have 3 compartments and black mesh on both outer walls. You raise the 2 inner doors and release a set number of moths into the middle compartment, then turn on a black light at a set distance. After 30 minutes, when the moths had settled, You close these inner doors and count the number in each compartment. In this example, of the 10 moths originally released into the tunnel, 3 never moved from the middle compartment, so we don't count them. Of the remaining 7, 6 moved towards the light and 1 moved away. 6 divided by 7 is 85% attraction. Stewart et al. Found that 87.5% of horned earworms moved towards a black light at 6 meters distance. They also found a lot of other things, but this number right up there is the only one I'm interested in. My question being, will horned earworms from urban and rural backgrounds today still move towards a black light 6 meters away 87.5% of the time? This project has taken 2 years, thousands of caterpillars, and 9 wonderful collaborators who manage sentinel corn plots spanning the horned earworm's overwintering range, shown there, which includes some very light polluted spots like Raleigh and Corpus Christi, as well as some very rural locations like Plymouth and Mac Run Ridge. We hand reared these caterpillars in the lab. This is not what hand rearing looks like, by the way. It looks like this, where you give them diet, but they just eat each other. So we hand reared these caterpillars in the lab to adults, through to a second generation of adults to impossible, although we had to test the first generation in many cases due to temperature limitation. And last summer, we took our populations to the Harvard Forest in Petersham, Massachusetts, where we set up light attraction trials as described in Stewart et al. 1969. Here are our 2 boxes with the 3 compartments and sliding inner doors. The black light is there in the middle. Here's what it looked like to the moths inside the tunnel. And again, we were looking to see how many of them moved towards it into the far compartment versus away from it into this compartment here. This summer, we built even more boxes, reared even more moths, and took them even farther from home to a remote cabin in Lakeville, Maine, which is one of the only places in the eastern US with pristine night skies. And that's what it takes to have an open area as dark as it was in 1969. So this is a video of the experimental procedure. You can see we prepare the box as the sun sets, and once it gets dark, put the moths in, let them rest, turn on the light, let them fly to it. That only takes 30 minutes, at which point you go through, collect everybody, and see who went where. Interestingly, Stewart et al. Collected their moths using blacklight traps. They didn't rear them. So this year we added an extra step where the night before each trial, we would release our moths into a gazebo and catch them again so that they were wild caught. The gazebo step brought with it some new challenges, including hurricanes and very fearsome guardians of the gate, but we got through it. And here's what we found. So starting last year, the collection sites are shown on the x-axis, and they're plotted according to the environmental light levels at the collection site within a 50-kilometer buffer. The y-axis shows light attraction percentage. 50% line in white represents no choice, no preference one way or another. Moths are equally likely to go this way or this way. We did find a significant preference towards the light, about a 66% attraction across urban and rural genetic backgrounds. So there was no difference there along the x-axis. However, this current average was different than the historical value of 87.5%. And my initial interpretation of this work was that, well, yes, of course, because as I said before, the horned earworm is highly migratory and well mixed. We wouldn't necessarily expect differentiation in space, but perhaps there's differentiation in time. Perhaps they are evolving as one because they're behaving very differently now than they did in the past. To make sure that we were seeing a real difference, we ran the experiment again with new moths at a pristine dark sky site and recovered the historical value of 87.5%. Almost exactly, actually, it's eerily close. Our gazebo step made no difference, by the way. So in white, these points show wild caught moths from our blacklight trap, and the solid dots are their counterpoints that we got from around the gazebo. So this suggests that it's not about the details of the experimental design, and it's not about the urban, rural, or historical background. It's not about the genes. It's about the environment. In particular, perhaps in the Harvard Forest, there were a few other light sources visible, although we certainly didn't see them, but they may have interfered with the movement of the moths towards our black light. I think a much bigger difference is the overall level of sky glow. That and habitat structure. So here, the hardwood forest on the left there's a lot of foliage, obviously. So the light reflects off of these leaves in interesting ways. Where on the right, we have this truly open, dark habitat, which may be responsible for this difference we saw. We're still digging into it. It's very new data. But let me just return to the main point for now. So it seems that just as threats to moths are rapidly intensifying, including light pollution, our very methods of understanding are breaking down because of light pollution, leaving me with more questions than answers. To keep moving forward, my laboratory is right now working on developing modern methods of monitoring moth activity that don't rely on moth stupidity, putting an ecosystem services price tag on light pollution that integrates its effects on pollinators and pests, and coming up with practical solutions to help humans and insects peacefully coexist for generations to come. But for sustainable lighting policy and advocacy to succeed, we need buy-in from the general public. And well, all of us here in this room love moths. Everyone loves fireflies. Fireflies can act as flagship species to inspire dark sky conservation efforts, as long as the sustainable lighting practices that work for them work for other species as well. If you've never seen fireflies before, let me introduce you. Each string of flashes captured in this photograph are advertisements produced by patrolling males. I'll show you a video of a male here. He starts in the sort of upper right, and you'll see him do a very stereotyped flash pattern, one J-shaped flash separated by 6 seconds. And the other thing I want you to notice is that after each flash, he hovers for a little bit as if he's looking for something. So flash, 1, 2, 3, 4, 5, 6. Flash, 1, 2, 3, 4, 5, 6. Lost that one. 1, 2, 3, 4, 5, 6. Flash, 1, 2, 3, 4, 5, 6. And there's another one. So hopefully you saw the hovering as well. The male's looking for something, right? And he's looking for a response flash, hopefully from a receptive female conspecific perched in the vegetation, and not the imitation from a predatory female in a separate genus who wants to lure him in and eat him. The exchange of flashes between male and female fireflies is called the courtship dialogue, and videos of this are fairly rare, so I want to show you one just as a treat. On the left, well, actually throughout, we're going to see fireflies. The species is Photinus marginalis, and in these species, the males do a single flash, and the females respond very quickly afterwards, sort of flash type situation, except that you'll notice the female flash is a little more nuanced. There's a little bit of flicker and flare to it. So there you see flash. Again, so the male's climbing towards the female and using this dialogue. She didn't answer that one, unfortunately. Using this dialogue to find her and to maintain interest, right? We often mark females in the field, which lets us observe these fun behaviors. So here, that female is mating with a male connected to her on the bottom there, and there's this second male wandering around flashing, trying to see if he can break into this in some way. They very rarely give up, male fireflies, in my experience. You can see there he goes. And in fact, if you're really lucky, you might see what we call a triple stack, where on the right there's one female firefly at the very bottom. You can barely see her. Male on top is actively mating with her, and then there's a male on top of him and a male on top of him, and they're all sort of trying to figure it out. So needless to say, the sex lives of fireflies are highly dramatic and highly visual. As a result of strong selection, firefly vision is very specialized. So fireflies only have 2 types. Actually, since it's 4:45, I might skip this part, but ask me afterwards. We'll do a whole thing about firefly vision and visual communication under light pollution. So where do I want to leave off then? So let's move into the lab. So now we're looking at firefly courtship under laboratory conditions, and we're interested to see how different colors and intensities of light change this dynamic. What are the behavioral effects, and what can we take from this in understanding a firefly conservation situation? So in the lab, I developed this method of filming courtships where there's two chambers of a compartment, which is shown on the right. The males are in the top chamber and the females are in the bottom chamber. And in fact, here's a still image. So males are in the top, the females are in the bottom. Because they can't ever find each other, they'll just dialogue back and forth for an hour while you do all sorts of things to their environment. And here is a video of Photinus obscuralis courtship. So you can see the male flash. The female in the middle of the circle there responds every time. Flash, 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 and response. Under a light, in this case a cool white light, the male keeps flashing, although not quite as actively. And the female, still in the circle there, goes entirely dark. Yes, and so here's what that looks like statistically. So in darkness, male fireflies in these chambers flashed about 10 times a minute, and females responded almost every time. Under all 5 colors and both brightnesses of artificial lights, we saw male flash rates fall by about half, but the female response rates truly plummeted. To the point that under a bright amber light, we never saw a female flash once. There are many interesting implications to this, especially regarding how we interpret firefly conservation in the field. If we see a lot of males flashing under a streetlight, we think that they're fine, but they might just be wandering around looking for the females that aren't answering them. But also what's interesting to me is that on the bottom there, you can see that dim blue and dim red light are relatively less disruptive, which suggests a possible way forward, a sustainable lighting solution that we can take out into the real world. And so the last thing I want to do today is just that, take these ideas out of the laboratory and into the real world. Male fireflies in the genus Photinus carolinus flash in synchrony, all together 5 to 8 times. And then they wait for about 8 seconds of darkness, giving the females time to respond. And then the flashes come back again in a minute now. There they are. The video does not do it justice at all. It never does, but especially in this case, you really have to see this in person. And so these displays attract people from all around the world to remote parts of the United States, including Kellettville, Pennsylvania, home of the Pennsylvania Firefly Festival. And just for reference, in case you're curious, there's where the Harvard Forest is, and that's where Lakeville, Maine is. It's very dark. So other firefly festivals, which I will not name, don't attempt to regulate the lights that visitors bring when they come to see fireflies. Of course, visitors have to see where they're going for safety reasons. These events happen in the woods, there are unpaved trails, so visitors probably should have access to some kind of light. But the Pennsylvania Firefly Festival is trying to find a middle ground that keeps people safe and fireflies happy. For decades now, firefly researchers have advocated the use of firefly-friendly light sources when out in the field, meaning lights of a color that fireflies can't see very well or don't react to. We just can't agree on what that color is, red or blue. Amber is a third option, which is often touted as a generally insect-friendly light because it attracts fewer moths and other insects than a black light or even a blue light. So in 2019, we set out to explore the relative costs and benefits of these different lighting approaches in the following way. These are the grounds of the Pennsylvania Firefly Festival. The event happens in the woods up near the top. We worked along this forest edge here, where fireflies from the woods spilled out into the yard. We set up 4 plots with a 6-foot pole in the middle of each, from which we suspended shielded LED floodlights of varying color: blue, amber, red, and a dark control. They were all equally bright, except for the dark one, and the order of the treatments was randomized each day. Here's another overhead view. You can see the line of the 4 plots following the forest edge. We focus on one of these plots. You can see we divided it into 4 subplots, extending perpendicularly back towards the woods. Photinus carolinus are active for about 2 hours each night. So for 2 hours, myself and my research assistant, Caroline Dresler, would walk these transects, counting the number of male flash patterns we saw. We also regularly sampled other insects swarming around each of our lights using sticky traps replaced at intervals. Before I show you the data, let me, for the final time, I think, orient you to the axes. So the x-axis shows the four subplots extending out from the forest edge on the left. Botanus carolinus is a forest species, but as I said, there were so many males that in the woods that they spilled out into the yard, and we saw a pattern that looks like this. That in darkness, there were the most flash patterns per minute near the forest edge, and it sort of fell off as we went out into the field. When we put an amber light right in the middle, we saw a huge drop in male courtship flash activity and similar but less dramatic drops under blue and red light. All three colors significantly impacted the firefly display in this area. None of them did anything over here in the forest, and only blue and especially amber had an impact out there in the field. So to summarize, similar to the previous experiment, amber is the worst and blue and red seem to be relatively okay as far as the fireflies go, although of course darkness is best. And now to add back in the flight to light dimension. So the sticky trap data we got looks something like this. We did see a few insects on our sticky traps in the dark that were unluckily merely passing through. Under the blue lights, we caught a lot more insects, and unexpectedly, we got a lot under the amber lights as well, equally much statistically speaking. And then in the red lights, it was about half as the blue and the amber. This was unexpected because we would predict most insects are most attracted to blue, and it might have something to do with the composition of the community we were sampling, which was very mosquito, gnat, fly heavy, and there were actually quite few moths in that area. But the thing to take away is that in this scenario, red is pretty good, and amber is no better, in fact, equally bad as blue. So putting these things together, we see a little bit of a solution. Seems like red is pretty good, amber not so much. Wouldn't it be a shame if amber lights were becoming increasingly popular as dark sky friendly, sustainable lighting solutions? We've spent many years in my field trying to find a magical color of light that only humans can see, that has no serious astronomical or ecological impacts. But by now, most of us have realized that while we might be attracted to lights like these, and in fact, you might make the acquaintance of a number of dark sky conservationists at this one, they are distracting us from better alternatives, highly metaphorically. Several years ago now, I worked with Xerces to come up with a set of firefly-friendly lighting guidelines that de-emphasize color in favor of more universally effective mitigation methods, which I listed in order of priority. That's as I saw it then. Today, while the recommendations themselves have not changed, their relative level of priority has, as we've come up with more evidence about what is more or less beneficial for insects generally. It's something I want to pontificate about a lot in this recent perspective piece on the bottom there. But for now, I'll just show you the recommendations as I order them currently. First and foremost, we ask that people remove unnecessary lights. This includes purely aesthetic lighting, tree lighting, string lighting, etc. Those lights that they do need, we ask people to install motion activators so that they're only on when people are there to use them. We ask them to shield their lights to the smallest acceptable area, focusing on a sidewalk instead of a yard. To dim them as low as they can live with, given that super bright LEDs are sort of raising the temperature, so to speak, of the outside constantly, but you would be surprised how much you can see with very little light. And then we ask people to make them as red as possible, knowing that human behavior comes into play here. Some people think red lights are just a bit too spooky, so you have to do what you can and compromise. And then finally, we do suggest timers, although I think now they're probably a lot less useful than people might think. The number one recommendation, though, is that people turn out their lights when they go indoors, especially in natural areas, so that we can restore these ecosystems to their natural state, which is darkness. And by doing this, we can support the fireflies, moths, and other creatures that call them. So with that, I would like to thank my committee, my collaborators, my research assistants, and my funding sources. These are all folks who are involved in my PhD research. And here is my current lab, as well as collaborators. Students on the top right, in the middle, these are the folks that worked with me on the first core and earworm project I talked about, and on the bottom, the second core and earworm project. And I'm happy to take any questions. Yeah.
A super cool talk. So in my PhD, I studied nocturnal insects, and we always used red light because insects can't see red light. So my question for you is like, is that not really true? Or do you think like the LED light, there's some like a little bit of bleed of like a wavelength that they can see? Or yeah, I'm wondering like how your results kind of fit into that narrative.
Yeah, fireflies have a simple version of insect vision. So most beetles are missing a blue photoreceptor. So you can imagine one, but fireflies are a good example because they're simple. So they have one UV photoreceptor and one green photoreceptor. And so yes, they don't have a red photoreceptor, right? And that means they're not very good at seeing red. It also means they're red-green colorblind. However, like so many things, this is a probability distribution. And so a green photoreceptor can see red light, it's just not as good at it. So it has to be brighter in order to be picked up. The thing is, so many red LED headlamps are incredibly bright. And in fact, when we're out in the field, we use layers and layers of just paper to make them dim enough so that the fireflies don't seem to mind them. I definitely think it's an approach, but it's not a perfect solution.
Amazing job. People often in certain like urban areas complain about streets being too dark for like human movement and humans feeling safe. I'm just curious if what the dialogue is of people being like, they should put streetlights in because I don't feel safe walking at night versus the safety of the insects.
The other day, a cyclist coalition in Arlington was advocating for more lights on the bicycle paths, and I felt so conflicted as a cyclist because I get it, and especially in terms of driving, cycling, or walking, you need to have an awareness of your surroundings for safety reasons. That is totally true. And I don't think we need to convince people to walk through dark alleys as a losing battle. And that's why I emphasize the focus on natural ecosystems, right? If your house is next to a park, I think you need to think very seriously about this. If you're in an apartment complex in San Francisco, it's probably your light won't really make a big difference in the scheme of things. So I wouldn't worry about it too overly much. In terms of long-term future utopia solutions, motion activators solve this problem. When you're there, the light's on. When you're not, it's not on. And that's like really all it comes down to. Yeah.
At the start of the talk, you mentioned some things about ecosystem services and kind of weighing the pros and cons of these different ideas about light. And I'm wondering if you could talk just in broad strokes about what sorts of things you're thinking about as like pros of having light and cons of having light and how you're going to compare those things.
Yeah. So I think it's interesting to study the effects of light on a pest species, because if we show that it's bad for the pests, a lot of people might think, isn't that great? And in fact, light is used in pest control in many cases. Greenhouses will control their lighting regimes in order to suppress feeding activity at night, for an example. So I think that's worth thinking about. So often though, it's this very similar insects are living in the same place. Some are doing the good thing, some are doing the bad thing. And so I'm on the search now for these paired systems in which we can investigate both pollination and feeding activity on crops of relevance. And I think there's a lot to dig into there, yet we are missing a fundamental first step, which is documenting nocturnal pollination in its natural context. We really don't know a lot about what's visiting flowers at night, and I think it's an exciting new field that I hope to get into. Yeah.
This was super cool. I feel like folks who work on sea turtles talk about light pollution, like policy all the time. And like, obviously those two things, I mean, well, I guess insects are in the same places as sea turtles, but sea turtles are often not where insects are. I'm just wondering, like, if like people who work on these, like the same issue in different systems, like if you've like talked to those folks much and if like similar policies work. And sea turtle folks use red lights for all our.
Absolutely. No, that's a really good question. So yes, for a long time, talk about the ecological impacts of light pollution. Number one, there wasn't any. For many decades, this was an astronomical issue only. In 1976, they published an article in Science about how lights were interfering with astronomical observations, and it took 30 years for somebody to talk about what it might be doing to animals. So that's one big thing. A lot of the policy and advocacy work that we have so far has come from astronomers, and it's been very astronomy focused. That's why we talk about shielding. Red light is particularly good because it doesn't create sky glow as much as blue light. And so red light is a focus, shielding and timers and so forth. From the ecological side of things, the turtle people definitely predate the insect people by quite a while. And folks studying bird collisions as well. There's a lot of work there. We are all on the same team, and I definitely do talk with people working in other systems. And I think we can identify common solutions, and they're really simple, of course. What I like about studying insects, though, is I think insects are uniquely affected by artificial light